A review of a novel role for xanopsin, stimulation of food intake. Xanopsin is taken from skin extracts of amphibians. It is not clear what role xanopsin plays in amphibians, but it does affect the digestive system of other animals. In dogs, it stimulates pancreatic secretion and inhibits gastric excretion. Xanopsin has an identical amino acid sequence to xenin, which has been shown to inhibit food intake in animals and increase activity in the ventromedial hypothalamus, which is associated with perception of being sated. Because xanopsin and xenin have similar structure, the researchers hypothesized that xanopsin would have the same inhibitory effects that xenin is shown to have. The researchers performed a freehand intracerebroventricular injection of the xanopsin. The chick was held in a restraining device that left the head open for injection. They targeted the left lateral ventricle. The xanopsin was dissolved in 5 microliter artificial cerebrospinal fluid solution with a blue dye to facilitate localization. For the first experiment, the effect of xanopsin on food and water intake was tested. The chicks fasted for 3 hours and then randomly got injected with different amounts of xanopsin. After injection, the chicks returned to their individual cages with free access to food and water. Food and water consumption were measured every 30 minutes. For the second experiment, they repeated the first experiment, but did not allow the chicks to fast before injecting them with xanopsin. The third experiment was identical to the second experiment, except that the doses of xanopsin given to the chicks were higher. The fourth experiment was to test the expression of CFOS in the hypothalamus. The fifth experiment was done to test the effect of xanopsin on 16-day post-hatch chicks. This time, a guide cannula and a microsyringe were used to administer the xanopsin to the third ventricle. The sixth experiment was identical to the fifth experiment, except a higher dose of xanopsin was used. In the seventh experiment, the researchers randomly assigned chicks a small dose of xanopsin, anesthetized the chicks, and then decapitated them so they could find the expression of the appetite-associated mRNA. The eighth and final test was to see the behavior of the chicks post-injection. They injected small doses of xanopsin into the chicks and moved them into open areas by themselves with food and water. The chicks could not see other chicks, but could hear them in order to reduce isolation stress. The chicks were monitored using 3 cameras, and every movement from pecking, perching, and preening to pooping was tallied. In every experiment, some chicks were injected with a solution without xanopsin to create a control group. After the data was collected, the chicks were decapitated, and its head was cut open to determine the site of injection was correct. The results from experiment 1 showed that the xanopsin did not affect food or water intake after the chicks fasted for 3 hours. In experiment 2, the chicks that were injected with 0.3 and 1 nanomole of xanopsin increased food intake 30 minutes after the injection. The chicks injected with 3 nanomoles of xanopsin increased food intake from 30 minutes to 150 minutes after the injection. After the full 3 hours, the effects had fully diminished, and there were differences among the chicks with different concentrations of xanopsin. Xanopsin did not influence water intake for the full 3-hour period. In experiment 3, the chicks injected with 1 and 3 nanomoles of xanopsin increased food intake throughout their entire 3-hour period. The chicks injected with 9 nanomoles decreased their water intake for the first hour after the injection. In the fourth experiment, chicks with xanopsin had varying degrees of CFOS expression in the hypothalamus. CFOS immunoreactivity increased by 150% in the lateral hypothalamus in the chicks with xanopsin compared to the control group. In the fifth experiment, the chicks with 0.3 nanomoles of xanopsin increased food intake from 30 to 45 minutes. Food intake increased at 45 minutes for the chicks injected with 1 nanomole of xanopsin. In experiment 6, the chicks with xanopsin increased food intake. The one with 0.3 nanomoles of xanopsin increased intake from 30 to 75 minutes. In experiment 7, the chicks with Xenopsin increased expression of POMC mRNA through the entire hypothalamus, but the amounts AGRP and neuropeptide Y mRNA were not affected. In the final experiment, the chicks with Xenopsin increased their number of feeding pecks between 20 and 60 minutes. These chicks also spent less time sitting from 35 to 60 minutes and spent less time in deep rest, eyes closed for over 3 seconds, from 45 to 55 minutes. The researchers' original hypothesis was that Xenopsin would decrease food and water intake, but the results from experiment 1 do not support this. Even after a 3-hour fast, the food intake was not altered. This fast is common in experiments where an anorexigenic effect is expected. When an increase of food intake is expected, a fast is not commonly part of the experiment. This is how it was in experiment 2. This experiment showed that Xenopsin increased food intake, with 3 nanomoles being the most effective dose. Experiment 3 shows us that higher doses are not better. The chicks that were injected with 9 nanomoles of Xenopsin did not increase food intake and actually decreased water intake, which the researchers speculated was due to the malaise in the chicks. These experiments show that there is a narrow range of doses that increase food intake. These doses are notably higher than doses of other chemicals that increase food intake, such as neuropeptide Y. Experiment 4 determined that the lateral hypothalamus was associated with Xenopsin's orexigenic effects, which is conducive to the theories of its role in hunger perception. Experiment 5 showed that the dose-response duration was smaller in older chicks. The response lasted for 150 minutes in the 4-day post-hatch chick, while it only lasted for 45 minutes in the 16-day post-hatch chicks. The stimulation of food intake in experiment 6 supports the idea that the lateral hypothalamus has receptors for the orexigenic response. The response duration was shorter in experiment 5 than experiment 6. This may be because the lateral hypothalamus is the furthest appetite-associated hypothalamic nucleus from the third ventricle where the injection was administered. So because experiment 5 had a lower concentration of Xenopsin, it took a longer time for the peptide to diffuse into it. Because of previous experiments, the researchers know that CFOS immunoreactivity was not affected by xenin, and when xenin was injected into the lateral hypothalamus, there was no effect on food intake. Therefore, xenin and Xenopsin affect food intake through different mechanisms. Xenopsin may act alone or work with other chemicals. That's why experiment 7 measured mRNA abundance of the most well-known appetite-associated neuropeptides. POMC mRNA was found, but is not clear if POMC or one of its derivatives acts with Xenopsin. It is also unclear if any lateral hypothalamus projections involved regions outside the hypothalamus to stimulate food intake. The behavioral analysis in experiment 6 shows the primary effect on Xenopsin is on food intake because it only increased the amount of food eaten and the amount of feeding pecks while decreasing other behaviors like sitting and deep rest. In conclusion, these experiments provide evidence that intracerebroventricular Xenopsin has an orexigenic effect on chicks. POMC or its derivatives could aid in this. The response is dependent on dose, with higher doses causing malaise. These findings could be of help in the medical and agricultural communities. Because Xenopsin causes increased food intake, it could help people who suffer from anorexia. It could also make animals that are raised for consumption eat more and increase their body mass. If I were to recreate the study, I would do a couple things differently. In experiments 4, 7, and 8, only one concentration of Xenopsin was used along with the control. I would use at least 3 different concentrations to determine a dose-response curve like they did for the other experiments. Also, now that there is more evidence that the lateral hypothalamus is responsible for the orexigenic properties, I would inject the Xenopsin directly into it instead of the third ventricle so that time it takes to diffuse into the lateral hypothalamus doesn't skew the results.